Part 2 of the human defence system, where we deal with specific immunity. This is geared towards the Irish Leaving Cert, but it does include added material, so please refer to your textbook. Specific immunity. This involves two types of particular white blood cells. They're both lymphocytes. We have B lymphocytes and T lymphocytes. So B cells and T cells. B cells are produced in and mature in the bone marrow. They then migrate to the lymphatic system. B cells are the only cells that produce antibodies. You have thousands of unique B cells, and they are unique because each one of those B cells is covered in one type of antibody. There is a particular B cell with a particular antibody on its surface that will recognize a specific antigen. No other B cell will have that same antibody that will connect with that antigen. So each B cell is highly specific. T cells are also produced in the bone marrow, but they mature in the thymus gland, and that's located in your thoracic cavity. From there, they migrate to the lymphatic system also. There are 4 types of T cells. There are helper T cells. These are the most important of all the immune cells. Then we have memory T cells, killer T cells, and then suppressor T cells. T cells have antigen receptors on their surface. Each T cell has only one type of receptor. That receptor will recognize one particular antigen. So T cells are also antigen specific too. So just like the B cells, they only recognize a particular type of antigen. And it's really important that you make the connection that both B cells and T cells are recognizing a specific type of antigen. There will be different B cells and different T cells for different antigens. B cells will deal with pathogens in the fluid of the body, bodily fluid like in the blood and in the tissue fluid. And T cells, such as killer T cells, they deal with already infected or abnormal cells like cancer cells. So let's tell the story then of the specific immune response. A pathogen enters the body and immediately a macrophage engulfs and destroys this pathogen. Now, once the macrophage has destroyed the pathogen, it takes the antigen, that foreign molecule, off the pathogen and puts it on its own surface. So the macrophage presents that foreign antigen. A helper T cell with the correct receptor on its surface, the receptor that matches that antigen, attaches to it, and this immediately activates that helper T cell. This activated helper T cell will multiply, forming many identical copies or clones of itself, and some of these will be memory T cells, always around to remember that antigen and to help switch on the specific immune response to that antigen if they ever need to again. These activated helper T cells, they also secrete chemicals, and these chemicals act as messengers that call the killer T cells and all the other cells into action and tells them to up the ante and to basically produce more identical copies of themselves. These killer T cells, so these are the guys that do the killing, they will kill damaged or infected cells. How do they do that? Well, killer T cells, they secrete perforin, and these are proteins, and these proteins get projected or inserted into abnormal or infected cells, basically piercing them, and they fill up with water, and these abnormal or infected cells will eventually lyse and burst. But why do they do that? They don't do it to normal cells, only abnormal or infected cells. And the reason why I know that they're abnormal or infected is because they recognize that antigen. They have receptors on their surface that help them to recognize that foreign antigen. So if a B cell then with the correct antibody specific to that antigen comes into contact with the pathogen in the fluid of the body, it binds to that antigen, and this half activates the B cell. So the B cell is covered in antibodies, and these antibodies identify that antigen, and the B cell connects onto that pathogen, connecting at the antigen. So the B cell at this stage is only half activated. It really needs the chemicals from that activated helper T cell and also the helper T cell itself to come and attach onto the surface of the B cell before the B cell becomes fully activated. Once this happens, so once the chemicals from the activated helper cell and once the activated helper T cell connect onto the surface of the B cell is fully activated. It divides many times, forming loads of these plasma B cells and memory B cells. The plasma B cells are the antibody producing cells. They secrete huge quantities of the correct antibody, so they flood your system with these antibodies that are basically matches to that antigen. So when the pathogen is overcome, the plasma B cells will die off and you'll have memory B cells that will survive, ready with the know-how on how to make that exact antibody if they need to again. Then you have the suppressor T cells. So once the whole fight is over and you've overcome that pathogen, the suppressor T cells call off the response. The plasma B cells die off and the activated helper T cells die off, just leaving those memory T cells and the memory B cells. So this was your primary immune response, and this can take between 10 and 15 days really to master how to achieve B cell activation. The primary immune response is slow, but the secondary immune response will be faster and stronger. Why? Because you have those memory T cells able to recognize that antigen and to stimulate those memory B cells to divide and form the correct plasma cells all over again, ready to secrete lots of that antibody specific to that antigen, and they do it so fast and in such great quantities that you don't even become symptomatic. You don't even show symptoms of the infection. So just to note that antibodies do not kill pathogens. What do? Well, they attach to the surface of the pathogens, they attach onto their antigens, and they prevent them from entering into cells. They cause them to clump together as well, and this basically makes it easier for the macrophages to engulf and destroy them. So that was the specific immune response. I hope the video helped. Remember to go over all your definitions and to use your textbook, and I'll put a little third video up describing the different types of immunity as well. Best of luck.